Omics, Omics Peak Part 1. Omics Peak because of the immediately attractive upswing created by the genomics revolution and the large financial resources made available in many industrialized countries. Adjacent fields of science have adopted similar terms, leading to a great proliferation of designations by such as transcriptomics, proteomics, and metabolomics. Some biologists have complained that what was molecular biology before is now named after one of the omics, but in fact is still molecular biology. Zhou et al. 2004 proposed a classification of genomics according to the three main categories: approach, structural or functional, scientific discipline, evolutionary genomics, ecological genomics, etc. Objects of study: plant genomics, microbial genomics, etc. There are obvious terms such as pharmacogenomics, cardiogenomics, awkward ones such as saccharomics, the study of all the carbohydrates in the cell; vaccinomics, the use of genomics for vaccine development. The three most common extensions of genomics are transcriptomics, proteomics, metabolomics. The relationship between genomics, transcriptomics, proteomics, and metabolomics. The genome, DNA, transcription, RNA splicing, RNA editing, and the transcriptome, rRNA, mRNA, tRNA, and translation, and the proteome, enzyme, structural proteins, transcription factors, ion channels, signaling proteins, and receptors, catalysis, synthesis, transport, transformation, the metabolome, and the studies metabolomics, the proteome, and the studies proteomics, the transcriptome, and the studies transcriptomics, the genome, and the studies genomics. And sugars, amino acids, lipids, secondary metabolites, hormones. Yeast, fly, worm, and weed. Part 4. Diversity of physiologies. The new range of models should embrace diverse phylogenetic lineages varying in their physiology and life history strategy. For example, the model plants Arabidopsis and rice both employ the C3 photosynthetic pathway to complement our genomic knowledge of primary production. Genomic analysis of plant utilizing C4 photosynthesis, sorghum, or crassulaceous acid metabolism (CAM) will be highly informative. Plants utilizing C4 photosynthesis or crassulaceous acid metabolism, considering the diversity of life histories, species differing in their mode of reproduction and dispersal capacity, should be chosen. For example, hermaphroditism versus gonochorism, parthenogenesis versus bisexual reproduction. Species ecological interactions. Species that take part in critical ecological interactions, mutualisms, antagonisms, are obvious candidates for genomic analysis. One may think of mycorrhizae, nitrogen-fixing symbionts, pollinators, natural enemies of pests parasites, etc. The most obvious strategy for analyzing such interactions would be to sequence the genomes of the players involved and to try and understand interactions between them from mutualisms or antagonisms in gene expression. Suitability for field studies. The wealth of knowledge from experienced field ecologists should play a role in deciding about new ecogenomic models. Not all species lend themselves to studies of behavior, foraging strategy, habitat choice, population size and age structure, dispersal or migration in the field simply because they are too rare, not easily spotted, difficult to sample quantitatively, impossible to mark and recapture, not easy to distinguish from related species or inaccessible or too invasive techniques. In infrastructure, large active and interactive community of investigators, physical and virtual community resources, interaction with other basic and applied communities. Ecological context. Relatively undisturbed habitats in the native range of the species, observable ecology and behavior in nature, genetic differentiation causing local adaptation to a range of abiotic or biotic environments, legally protected field sites for long-term ecological studies, variation in sequence and phenotype, nucleotide variants in natural populations, abiotic and biotic environmental factors created with each segregating haplotype, evolutionary forces underlying nucleotide variation inferred from molecular evolution analysis, characterized phenotypes under natural conditions for each variant, impact of variants on fitness, abundance, range, and persistence known, structure and dynamics of the natural population known, molecular data access to genomic sequence and chromosomal maps, upstream regulators and downstream targets identified for the gene of interest, function of gene product known and its impact on fitness under natural conditions inferred, gene discovery and phylogenetic data, forward and reverse genetic tools, capacity to detect variation including differences in transcript and protein levels, known physiology to enable, for example, historical change in traits of interest to be inferred. This was ideal model species. DNA microarrays. All questions in ecological genomics require the full-length DNA sequence of a species before they can be answered. Some issues may prove to be solvable with the use of less extensive genomic investigations. For example, a gene hunt followed by multiplex quantitative PCR rather than transcription profiling with microarrays of the complete genome. Microarray studies with part of the expressed genome are possible even in species lacking a complete DNA sequence, microarrays can be manufactured at costs that are affordable for small research groups if they are limited to genes associated with the specific function or response pathway. DNA microarrays, with their associated massive generation of data on expression profiles, are one of the most tangible features of modern genomics and are often seen as holding the greatest promise for solving problems in ecology. Ecologists have two options. One is to develop their own microarrays, starting with spotted cDNAs of unknown sequence, doing a lot of tedious sequencing work and gradually finding out more about the genome of their study species. Another option is to apply transcriptome samples of known models to microarrays of model species.